Hello, I'm Patrick Dettinger, President and CEO of Absorption Systems, the drug absorption company. I am pleased to introduce a tutorial on transporter-mediated drug-drug interactions.
Let's begin with the FDA's position on drug interactions. In September 2006, the FDA released a draft guidance on drug interaction studies, updating a 1997 guidance on the same topic. A few key provisions are relevant to this tutorial. For the first time, the FDA addressed transporter-mediated in addition to metabolism-mediated drug-drug interactions. In particular, they're interested in a drug transporter called P-glycoprotein. One of the most important facets of the draft guidance is its implications for clinical trials. If the results of in vitro studies are positive for transporter interaction, you have to do a clinical drug interaction study. On the other hand, negative in vitro results mean that you do not have to do a clinical drug interaction study, which can save you probably 6 months, not to mention several $100,000. For now, the FDA is focusing on P-glycoprotein, or PGP, for two main reasons. 1. PGP is the best understood transporter and the only one for which some reagents are available to study it. And 2. PGP is present everywhere in the body that matters for drug disposition: the intestine, the liver, the kidneys, and the blood-brain barrier. The FDA has told us which assay format to use and how to identify substrates of efflux transporters. It just happens to be the assay that Absorption Systems was founded on more than 10 years ago. This leads us to the Absorption Systems solution, CellPort Technologies, a definitive assay system for drug transporters to determine which, if any, of the efflux transporters the drug is interacting with. Let's look at our approach. We start with a CACO2 cell, a human cell line, so we don't have to worry about dog PGP, and we eliminate one efflux transporter at a time. When we're finished, we will have a panel of at least 3 different cell lines, each of which is missing one transporter, in addition to the wild-type cells. This is the only way you can positively identify which efflux transporter your drug interacts with in the assay format the FDA has identified as definitive. So let's review. The FDA expects to see and will soon require in vitro data on PGP interactions, and has specified the bidirectional monolayer transport assay as the definitive format. Absorption Systems has a superior assay system, CellPort Technologies, that can be used in the FDA-specified assay format. It is based on a human cell line. It provides positive identification of efflux transporter interactions, and there is no need to rely on non-selective chemical inhibitors.
Thank you very much for your time. We look forward to helping you comply with the new FDA draft guidelines.